Hello, everybody. It's a great pleasure for me to be here with you. Let me introduce myself. My name is Pietro Quaglino. I am professor of dermatology at the dermatology clinic of the University of Turin in the northern part of Italy. And I'm here to share with you the preliminary results of this project that we could develop thanks to the great help and support by the Cutaneous Lymphoma Foundation. And the title of this project, I apologize for that, is a little bit difficult, as a mechanistic insight into the CD39, CD73 adenosinergic immune suppressive axis in patients with Sézary syndrome, association with disease course and treatment response. But basically, the main important point of this study that we try to address is to study the immune suppressive development in this patient. So as you know, Sézary syndrome show a difficult and aggressive disease course with a severe impairment in immune system. And we focus our attention on a small molecule which is called adenosine and the mechanisms through which this molecule is made. And definitely there are two different pathways, one related to CD39, CD73, and the other related to CD38. And we studied this pathway in the blood, so in the lymphoid neoplastic circulating cells and in the skin. And we found that the majority of patients show high levels of CD39 and CD73, and the minority high levels of CD38. And this is in the blood and the skin. And it's important, if we block these markers, there's a reduction in the adenosine. So we can reduce the immune suppression related to adenosine production by blocking these pathways. So this is important because we have the possibility to have monoclonal antibodies available that target these two pathways, so CD39, 73, and CD38. So the identification of this altered expression in this pathway could identify patients that could benefit from treatment with one or the other of these monoclonal antibodies. So it's a good point for the clinical management of this patient. And the other point is that we are trying to correlate this expression of these markers with the disease outcome and clinical characteristic of this patient, to identify if patients characterized with altered expression of one or the other markers show a different disease outcome and different response and different clinical behavior. So the results of these studies confirmed at the end of this project could induce an improvement in the management of this patient, identify patients that could benefit from specific treatment and reduce the risk of immune suppression. So we are definitely indebted to the Cutaneous Lymphoma Foundation for this possibility. And basically this work was possible thanks to your support and the support of the grant, and which was also very important for the development of the work in my institution and also my personal experience as researcher, because it gave us the possibility to study and to better understanding these processes, and so induce an improvement in the knowledge and clinical management of this patient. So many, many thanks again. And I'm looking forward to share with you the updated results and future developments of this study. Thanks again.
My name is Asya Angelova. I'm a virologist and work in the Clinical Cooperation Unit Viral Therapy of the German Cancer Research Center in Heidelberg. In our unit, we explore the potential of various viral therapy approaches, including oncolytic viruses against various types of cancer. By oncolytic viruses, we understand those viruses that are capable of infecting, multiplying in, and killing tumor cells without causing any harm to healthy cells and tissues. My team's research focuses in particular on the smallest oncolytic virus known, the H1 parvovirus H1PV. This virus has been studied for many years in our laboratory, and its remarkable multifaceted tumor suppressive potential has been demonstrated against solid tumors and certain types of blood cancer. The title of my project is H1PV-induced oncolysis and tumor microenvironment immune stimulation in a novel heterotypic spheroid model of cutaneous T-cell lymphoma. The project aims at answering the general question of whether parvoviral therapy may be applicable to cutaneous T-cell lymphoma treatment, and pursues three major scientific goals: to establish an in vivo relevant in vitro model of cutaneous T-cell lymphoma, to evaluate H1PV-induced oncolysis in this model, and to study some parameters of H1PV-induced immunogenic tumor cell death. The in vitro model of cutaneous T-cell lymphoma that we have established for the first time represents a novel three-dimensional system which includes, in addition to lymphoma cells, various cell types from the tumor microenvironment. We are generating heterotypic spheroids composed of tumor cells, endothelial cells, fibroblasts, or keratinocytes. These spheroids, in contrast to classical cultures, offer the possibility to analyze various cellular, virological, and immunological markers in the context of cutaneous T-cell lymphoma spatial growth and crosstalk with other cells from the microenvironment. Our pilot experiments with H1PV showed that some cutaneous T-cell lymphoma cells were very efficiently infected with the virus. These cells were able to support a productive virus infection. After their death, they released progeny variants that could in turn infect cells which survived the first round of infection. However, other cells were more resistant to the oncolytic effects induced by the virus. In such cultures, proliferation of surviving cells prevailed over oncolysis. And this is a scenario which represents one of the major therapeutic challenges in the clinic. We are trying to understand what is the cause for this resistance and whether it can be overcome through combining H1PV treatment with other approaches, for example, inhibition of the cellular pro-survival protein BCL2. Last but not least, it is important to say that the oncolytic viral therapy approach remains currently largely unexplored in the cutaneous T-cell lymphoma research. Only one study can be found in the literature, and this study dates back to the very beginning of the century. Furthermore, oncolytic parvoviruses have been tested against blood cancers of B-cell origin or T-cell leukemias, but never against cutaneous T-cell lymphoma. Our project has the ambition to generate the first preclinical evidence in this regard, and it is this important launching step that has only been made possible through the generous support of the Cutaneous Lymphoma Foundation. We are very thankful to the Foundation for having selected our project to receive the Catalyst Research Grant for 2021. This grant, and above all, the trust of the Foundation in our research, is what allows us and motivates us to work hard for a future clinical translation of a new cutaneous T-cell lymphoma treatment.
Good morning, I'm Patrizia Fuschiotti. I'm an assistant professor in the Department of Medicine at the University of Pittsburgh. I would like to thank the Cutaneous Lymphoma Foundation for supporting my research. My research is focused on studying the role of IL-4 and IL-13 in the pathogenesis of cutaneous T-cell lymphoma. In previous studies, we found that these cytokines promote tumor progression by inducing proliferation of malignant lymphocytes. In the project that is funded by the Cutaneous Lymphoma Foundation, we aim to study the effect of these cytokines, IL-4 and IL-13, in all cells of the tumor microenvironment of mycosis fungoides. And the aim of this study is to identify novel markers of malignancy that can be developed for novel treatments for the patients with advanced mucosis fungoides, and eventually to tailor therapies to specific patients. In this project, we are using cutting-edge technologies like single-cell RNA sequencing with in well-characterized patient samples. The patient samples are provided by our collaborator, Dr. Larissa Gesking at Columbia University, with whom we have a long-standing collaboration. The funding of the Cutaneous Lymphoma Foundation has been extremely helpful for us for generating results that validate our hypothesis. And our goal is to build up on this by enlarging the number of patients that will be analyzed and by developing the candidate biomarkers of these studies that eventually will be used in the clinic. So our goal then is to apply for larger grants and to obtain some NIH funding for future work for this project. So thank you very much.